Welcome back, guys. My name is Emmanuel C. Zilaba. In today's tutorial, I'm going to be discussing spermatogenesis. It's just a combination of two words, spermato and genesis. The word genesis means beginning. So if we join these two words together, genesis means beginning, and this is toe, and this is entire sperm, right? This is the spelling for sperm. So spermatogenesis means we beginning to make sperm. Or for short, spermatogenesis just means the production of sperm. How you as a boy, how you producing sperm right now. Now, when does spermatogenesis starts? Spermatogenesis starts in boys when they hit puberty. Puberty is just maturity stage. Mostly there is a range for puberty in boys, from 11 to 15. That is, for some boys they can hit puberty at the age of 11. Some at 12 years old and some at 15 years old. So as a boy, once you hit puberty, you start producing sperm from that day until you die. So right now, you are producing sperm. So if you watch this video, you will understand the processes involved in the production of sperm. Now, spermatogenesis occurs in the testes of every male. Now, you can produce sperm if you don't have a precursor. And that precursor is a stem cell we call spermatogonium. This stem cell is formed in the seminiferous tubule of your testes. There is a specific tube inside your testes that we call seminiferous tubule. It is within that tubule that you find a stem cell called spermatogonium, this stem cell. And it is a diploid, meaning it has two sets of chromosomes, 46 chromosomes. So, the processes involved in spermatogenesis require 4 basic steps. Step 1 is mitosis. Step 2 is called meiosis 1. And step 3 is called meiosis 2. Step 4, which is the last step, is called spermatogenesis. So let's explain it one at a time so you can get a clear understanding of how spermatogenesis occurs in your testes as it is taking place right now. Now, the first step, which is mitosis, we will firstly have our spermatogonium. And this spermatogonium will divide into 2 daughter cells. That's the first step, and we call that mitosis. Remember, whenever you're living from a single cell to 2 daughter cells, and you still maintain the same number of chromosomes, diploid number of chromosomes, we call that mitosis. These 2 daughter cells you produce, which are diploid, they are called, each of them, the first one is called A spermatogonium, and the second one is called B spermatogonium. What can happen is that we don't want, for the stem cell we use, the spermatogonium, the original spermatogonium we started with, we don't want it to be depleted. So we want to replace it. So this A spermatogonium will go back to replace the original spermatogonium that we used previously. This B spermatogonium will move from this compartment into another compartment. And whenever it leaves that first compartment into the second compartment, we change the name from B spermatogonium to something called primary spermatocyte. So you see, when B spermatogonium leaves this entire environment to this environment, we call it primary spermatocyte. In the second step, the primary spermatocyte divides into 2 daughter cells. But the problem is these 2 daughter cells, they are no longer maintaining the diploid number of chromosomes. They are now haploid. They maintaining 1 set of chromosomes. Each of the daughter cells carry half, 1 set. Now, So from primary spermatocyte to secondary spermatocyte, this entire process is called meiosis 1. You know, in meiosis, we're trying to reduce the chromosome number from diploid to haploid. So instead of 2n that will be maintained throughout, we now reduce it down to n. This is haploid. So in meiosis 1, we reduce the chromosome number from diploid to haploid. And these haploid daughter cells, they are called secondary spermatocytes. Step 3. These secondary spermatocytes will then divide. Will further divide. They will undergo meiosis 2. And when you divide secondary spermatocyte and produce another daughter cells, these daughter cells, they are called spermates. So these two secondary spermatocytes will divide into 4 spermates. When this process occurs, we call it meiosis 2. Now, the last step is that these 4 spermates will then undergo differentiation to produce our sperm cells. So this first spermatid undergo differentiation and produce sperm. In the process of differentiation, we're trying to produce the tail, the acrosome of the sperm, the neck. Remember in our video when we're discussing the structure of the sperm. So if you have a spermatid, the spermatid undergoes differentiation to produce sperm. But the process by which each of these spermatid undergo differentiation to produce our sperm cell is what we call spermatogenesis. So this is how we derive our 4 steps. Step 1, mitosis. We're living from spermatogonium to A and B spermatogonium. A went back to replace the original spermatogonium. B descend, and then it changes into primary spermatocytes. Primary spermatocyte further divides in step 2, that we call meiosis 1, and then it gave us our secondary spermatocytes. Each of the secondary spermatocytes divide through meiosis 2, and then produce our spermates 4. But remember, the spermates are also haploid. Each of these spermates undergo differentiation to produce our sperm cell. Before this thing can start at puberty, there is a gland in your brain called the pituitary gland. The pituitary gland can produce FSH, follicle stimulating hormone, and luteinizing hormone. Follicle stimulating hormone can travel in the blood. It comes directly to a cell in the testis called the Sertoli cell. It will stimulate the Sertoli cell to produce antigen binding protein. Now, when it produces antigen binding protein, the next hormone that we call luteinizing hormone, LH, is also produced on the pituitary gland. It will also travel in the blood. The blood will bring it directly to a cell in your testis called the Leydig cell. LH will stimulate the Leydig cell to produce testosterone. And testosterone is the hormone that drives the entire cell. In order to carry on all these processes, testosterone is the one that drives that entire process. But testosterone is a lipid substance, it's a fatty substance, and fat and water don't dissolve. Fat is lipophilic, and fat is also hydrophobic, it hates water. So because testosterone is a lipid substance, and it is not soluble in this environment for this entire process to occur, that's why the antigen binding protein produced by the stimulation of the follicle stimulating hormone helps to make testosterone less lipophilic so that it can be able to drive the entire process to produce sperm. I repeat, testosterone cannot drive the entire process of spermatogenesis because it is not water soluble. It can dissolve. The entire environment is hydrophilic. So because it can dissolve in that environment, it needs antigen binding protein to help it so that it can be soluble in that environment to drive the entire process. So this is how the process of spermatogenesis is happening right now. Thank you for watching. Please don't forget to subscribe to our YouTube channel.